All right, welcome to New York State IPM Programs Seminar Series. This is our last one for the year. Stay tuned for a new lineup of monthly seminars in the spring of 2022, spring semester. The schedule for today is for 40 minutes, starting here in a minute or two. We're going to have the presentation, and we'll have 10 minutes for questions and answers. And if you'd like to stay around, at about 12:20, we'll have a time where you can unmute your mic and talk and have a conversation with Eric directly. So I'd like to introduce our speaker today, Eric Clifton. Eric is originally from Chicago, Illinois. He completed his bachelor's degree at Augustana College in Rockland, Illinois. He did both his MS and PhD in entomology at Iowa State University with doctors Aaron Grassman and Aaron Hodgson. His projects included IPM issues, including Western corn rootworm and soybean aphid. Eric joined Ann Hayek's lab as a postdoc in the summer of 2017. He has worked on invasives like Asian longhorn beetle and is now primarily working on spotted lanternfly. I was able to hear Eric's presentation on this just a few weeks ago, and the work is really fascinating. And we're going to hear some things that are just happening now in his lab. So it's really up-to-date information. And all of this will be helpful as we try to figure out strategies to help manage spotted lanternfly. So Eric, if you'd like to get started now, we're ready to hear.
Great. Yeah, thanks, Brian. Thank you for the introduction, and thank you all for being here today to see some of this talk on spotted lanternfly. So there's the title slide again. I'll be talking mostly about biocontrol studies using entomopathogenic fungi, and a little bit more about searching for egg masses in and around vineyards, which is some work I did this spring. And I'm not going to talk too much about the spotted lanternfly itself. There's plenty of great other resources, including the New York State IPM website, if you'd like to learn more about this pest. But an article in 2018 argued the point that this might just be one of the worst invasive species in 150 years. And you're probably familiar with the spread, especially since it's recently arrived in New York. But if you consider its location in 2014, this map from the Ecolab shows the density and location of spotted lanternfly infestations. And so they originally isolated to Berks County, Pennsylvania in 2014, but about 4 years later in 2018 here, you can see that populations became more abundant and started spreading into New Jersey, Virginia, and other states. And then here is current year, 2021. This doesn't even show some of the other more recent findings, including Indiana. But you can see that Yellow County up near Binghamton, that is here in Ithaca, actually. And of course, we're all very concerned about spotted lanternfly moving into New York because we grow a lot of grapes, especially for wine. And so you can see sort of this map showing the wine regions in New York and the fact that lanternfly populations are already starting to invade the Finger Lakes, the Lower Hudson, and Long Island. And there is a real cause for concern because we've already seen firsthand just how much damage they can do to our vineyards. And so in 2018 and 2019, there were several vineyards in Pennsylvania that had been seeing heavy damage from lanternfly feeding, in some cases killing major transects in their vineyards altogether. And there's that infamous photo on the right there showing just these dense populations really depleting sap from these grapevines. And just a few photos of other host plants. The concern too is that they don't just feed on cultivated grapes, they also feed on wild grapes, which once you start to identify them, they're pretty dominant in the landscape, and you can see them even places around here in New York. So the left image there is showing native or wild grapes. The right there are some cultivated grapes. And the trouble is that some of their other major preferred host plants like tree of heaven, even oriental bittersweet, these plants are not only invasive, but they're also pretty well spread throughout the landscape. And so I often see bittersweet wild grapes growing sometimes up tree of heaven trees in the landscape. And so when you talk about maybe just removing tree of heaven, well, then you have to consider what to do about these other host plants that they feed on. And I also like to show some of these pictures and videos of the honeydew that they produce. So when they feed, they excrete the sugary sap that provides a substrate for these yeasts and sooty mold to grow. And the sooty mold could even block photosynthesis. So here's a video now. If you look closely at the right angle of the light, you can see just these droplets. That is copious honeydew raining down from the tree because the tree is just covered in lanternfly adults. And then the image there on the right that shows you this sort of white yeast that sort of grows around the bases of trees that have heavy feeding. And when you have all of this honeydew, this also seems to attract different hymenopteran insects. And so here's some videos showing these vespids on the left. These are like yellow jackets in larger hornets. And this will come into play later when I talk about the fungi. You also see lots of ants feeding on this honeydew there in the middle video. But in some cases, I also see bumblebees and honeybees also collecting this honeydew. And so this is important to consider the fact that these native insects are drawn into these habitats. And so if you were to spray or kill these lanternfly populations, we have to consider the potential non-target impacts on these other insects visiting the honeydew. And then just a little bit about natural enemies. Well, I'm mostly going to talk about fungi, but just to show you, there are some other natural enemies that attack lanternflies. We see spiders, we see predatory bugs, especially starting to see or hear more reports about these praying mantids attacking lanternflies. But you can see there's also this classic biological control approach, maybe using parasitoids to control them. And some other researchers are studying this parasitoid called Anastatus orientalis there on the top left, but they have to first test to see if it'll attack other native insects before they can release it. And so jumping into the outline now, I'm going to just sort of break down these different sections. And I'm going to start here talking about how I got involved with lanternflies and some of these epizootics that we saw. So in August 2017, we started to get these photos in our email about these dead lanternflies with this white stuff growing on it. And in spring or May 2018, we started to visit some of these field sites that had heavy lanternfly populations. And just digging through the leaf litter at the base of trees, we started to find more of these dead adults that died in the fall of 2017. And this was a entomopathogenic fungus that killed them. And this fungus was identified as Bavaria bassiana. And so this is actually a pretty well-known entomopathogen. And these are just pictures of different hemipterans or bugs that are killed by this fungus. So you can see Bagrata bug, there's brown marmorated stink bug that is on the bottom left, tarnished plant bug, even the kudzu bug on the bottom right is killed by this fungus, and sometimes also sees epizootics where populations sort of get wiped out by this fungus. And if you're not familiar with these entomopathogens, this is a nice little schematic that breaks down the life cycle of this fungus. It starts with infection through the insect cuticle, then it goes into the host to produce toxins, and then you get these blastospores or hyphal bodies. And then once the host has died, the fungus emerges and it produces more spores or conidia that can go into infect other insects. And like a lot of other fungi, the more rainfall, the more humidity, the better that the fungi do. So, you think of a wet season, you see more mushrooms, same kind of scenario here with these insect pathogens. And this is relevant because in the summer and fall of 2018, some parts of Pennsylvania had their wettest years on records. And the city of Reading had its most rain that it's ever had in 2018. And so in early October, I got this email from Heather Leach, who was the extension specialist at the time at Penn State University. And she was telling us that there was this location where there were a lot of dead lanternflies, like the entire population in the area was killed and they didn't know why they were dying. In the picture on the left here, this shows an example of the typical tree at this field site. There's only one live lanternfly in the photo. The rest were actually killed by this different fungus. This was a fungus we identified as Batcoa major. And this fungus is pretty cool because when it kills the lanternflies, it produces these structures called rhizoids. And so the pictures there on the top, they point to, they sort of look like cat whiskers or these thick white strands, and they actually attach the lanternfly to the surface of a tree or a leaf. And then once the fungus grows out of their body, it actually actively shoots off spores. And then those are released into the environment. They sort of catch wind and they could spread to infect other lanternflies and sort of roll through the population. And here's just demonstrating that this enormous spore production by this fungus. So there's one killed on the ground, and then I just highlighted here, you can see that sort of dusty white powder, that is the Bat-Coa major spores being shot off. But it wasn't just Bat-Coa in these field sites where we saw these epizootics. We actually saw both Bavaria bassiana and Bat-Coa major working together to kill these populations. And so these bar graphs are just sort of breaking down the causes of mortality in these field sites. We studied this in 2 locations. And we essentially found that like 90 or more % of the dead lanternflies attached to the trees, those were killed by Bat-Coa major. But if you randomly sample the cadavers that were on the ground and around these trees, it was like a 50-50 mixture of lanternflies killed by Bavaria bassiana or those killed by Bat-Coa major. And so we published this work in PNAS. We got to make the cover issue there. And that sort of rolled us into the next few years of research, starting to learn more about these native pathogens and how they affect this invasive insect. And so this is the next section of the talk. I'll be talking about the diversity of hypocrealine fungi. That's just sort of a taxonomic term for those type of fungi. So Bavaria bassiana, they all kind of have this white spore powder appearance. Characteristic spore balls, you can kind of see it in the left image here. But you can't really tell apart species just based on morphology. They can look very similar. There's maybe a dozen or more different species in this genus located in North America. And so there's this phylogenetic tree on the right here. This just sort of lists off some different species, and Bavaria bassiana is up here at the top. And for our studies, we wanted to sort of model this after previous work done on another invasive. And there's this study published here on the emerald ash borer. And so they collected and identified Bavaria fungi from this host. And in the process, they discovered there were 5 different genotypes. You could also say strains, I guess, but 5 different types or genotypes of Bavaria bassiana and 2 genotypes of a different species called Bavaria pseudobassiana. And they even tested these in lab bioassays and found that one of them was more virulent to the strain GHA. And you'll hear that strain again later, but that is used in some commercial biopesticides. And so the idea here was that maybe they could make a new biopesticide using one of these native strains. And that's an idea we had too. So starting in 2018 and through 2020, we would repeatedly visit different field sites in southeastern Pennsylvania. You can kind of see Philadelphia here on the bottom right. Here's Lancaster, and these are all kind of focused on Reading. And this is where we saw some of these epizootics in 2018. But we go and we collect these fungi when we find them, and we do the molecular work to identify them. And I'm mostly talking here about the work on Bavaria. And so between 2018 and 2020, we've identified 165 isolates, and all of them infecting lanternflies were one species, and that one species was Bavaria bassiana. But the cool thing about this is that within the one species, there are 21 different genotypes infecting them. And that's what you see on the right is this pie chart with all these different genotypes. But 3 of the genotypes, and I'm just coding them with letters of the alphabet, A, B, and L, these are the most prevalent. And you can see these are the bigger chunks of the pie on the right side. So we found A, B, and L were the most common. And another fact is that we've only found genotype L from lanternflies. We've actually not found it killing non-target insects. And so we're also trying to get a better understanding of, well, what other native insects are being killed by this fungus? Maybe the fungus is passing from them to the lanternflies or vice versa. So this is just sort of a launch pad to see what's going on in these invaded habitats. And so just another point too is that we're not going to find every single non-target, but anytime we're collecting these dead lanternflies, we collect all dead non-target insects that we can find. And in the process here, we found, we found some yellow jackets, some flies. You can see an ant moth and this beetle. These were all killed by Bavaria bassiana. And some of them you can see on the far right, these have overlap with the strains we see in lanternflies. So you can see like strain A, for example, killed these 3 different insects. We did find a different species, Bavaria brongniardii, killing a yellow jacket. So the point here is that these different species are out there in the landscape, but we have not yet found this species like brongniardii infecting lanternflies. And then if we just look at the molecular database to get a better understanding of similarities to other hosts, we find that a lot of these isolates are very similar to those that kill emerald ash borer. Genotype L actually matches to isolates that kill bark beetles. So we see sort of similarities to other strains that kill forest insects. Genotype I, you didn't see it in the pie chart, but this one's actually matching to this strain, ATCC 74040. This is actually used in another product called Naturalis. And I'm actually going to talk about that again too later. But in the process of doing all this work, we have actually found some other very different fungi infecting them. So here's some photos and some more details. This green fungus, this is Metarhizium pemphigae. This is an interesting fungus. It has these sort of green spores. If you look under the microscope, there are these palisades of conidia sort of stacked end to end. And it's interesting because it's only really been found infecting a few hosts. It's been found on an aphid in England. It's been found infecting termites in Ontario, but otherwise, it's only really been found in soil samples. So as far as we know, this is the first case of this fungus infecting a large plant hopper like the spotted lanternfly. And then here's another very, very cool fungus. This is a fungus that we actually named and identified. This is Ophiocordyceps delicatula. And it produces, you can see on the top left image there, sort of these long lavender tentacle-like structures just growing out of the entire body. And we can even sometimes get those little structures to grow on agar cultures. And so to really verify if this is a new species or not, we had to use 3 different genes in PCR, put together the sequence data with other reference isolates on GenBank, and just to confirm that this is a new species and not just something similar here. And I'm not going to go into details about the taxonomy, but there's been a lot of revisions to fungal taxonomy. So some of these species say Hirsitella, some are Ophiocordyceps. Basically, you just need to know that Hirsitella is being suppressed in favor of this genus, Ophiocordyceps. But here's our new species here. It falls in this clade called the Hirsitella citriformis clade. And this is actually a group of fungi that infect other hemipterid insects, including here the Asian citrusilids. So this sort of purple fungus with the same structures, infecting another invasive plant hopper. So that was very exciting that we found this whole new fungus infecting spotted lanternfly. Okay, now I'm going to switch more to the applied side about biopesticides and do we have some products that we can use to manage this pest? So in 2019, we collaborated with Penn State University. There was this park near Philadelphia that had this heavy infestation of spotted lanternflies. And so people are using it every day. They didn't really, I don't think they would feel comfortable spraying some kind of chemical insecticide. So this was the perfect opportunity to test a biopesticide. And so here we tested this product called Bottega, and that contains Bavaria bassiana strain GHA. And it has a 0-hour pre-harvest interval. So you could basically spray this right up until you harvest a crop if you're using it like on grapes or something else. And so we set up these split plots throughout the park. You can see they're sort of along walkways, and these sort of these buffers or these strips along the paths, these were loaded with tree of heaven, bittersweet, and other preferred host plants that lanternflies use. And we sprayed twice. We sprayed the nymphs in early July, and then we sprayed again to treat the adults in mid-August. And so here's photos there of John Rost from Penn State spraying water or Bottega to these plots. And then here's what we did to test the efficacy. For the nymphs, we did visual counts. Actually, at the time in 2019, we still didn't really have a good strategy to assess efficacy. So we sort of adapted this from work I've done on aphids and other pests where you do in situ visual counts. So these are bar graphs showing the numbers per plot. We did timed visual counts with multiple counters to get the average. And so we have the numbers here, no difference in the control or what would be the treated plots before spraying. But we found that there were less lanternflies in the treated plots 12 and 14 days later. And I'm just pointing here that the numbers in the Bottega treated plots were about 48% lower than the controls. And this was 2 weeks after we sprayed. And to assess sort of the persistence of the fungus in the environment, we collected leaf samples after spraying. These leaves were washed and the rinsate was plated on these selective media plates. So you see here, these are the Bavaria fungi growing on plates. You carry out the math and you could figure out the density of the fungus on a leaf area. And then we use this nice app called Leaf Scan to measure the area of the leaves that we washed. And basically we just found that the fungus lasts for about 5 to 7 days before it basically dissipates entirely from the environment. And this is a result I'm more confident in. The visual counts, we're not really sure if that's something to use in the future, but I like the approach we used here when we sprayed the adults. So what we did is we sprayed the plots again. And 1 day after the plots were sprayed, we went in and randomly sampled adults from those trees. We brought them to the Berks campus nearby and we reared them on these potted tree of heaven plants kept in a shade house. And so they're still exposed to the weather, the rainfall that's occurring in the area. They're not thrown into some kind of growth chamber. And so what we saw, this is survival on the y-axis, the days after sampling. Didn't see a lot of mortality. There was a heat wave, days 3 to 7 after the spray. But once it cooled off, we started to see more of these lanternflies die. And 14 days later, about 52% were dead. So 52% of the adults we collected died. And pointing here to this picture, among those dead lanternflies from the treated plots, 83% had fungal outgrowth. So this helped us to verify that they are dying from the fungus and not from other causes. And I use that word environmentally safe. Well, what happens to the other insects in these areas when you spray? And to assess this in August, we laid out these ground cloths you see here. We set these up around 4 trees in each plot. And every couple of days, we would go and we collect every single dead insect we could get off of these ground cloths, put them in humid containers to allow for fungal outgrowth. And even if a fungus grew out of it, looked like Bavaria bassiana, we had to verify that it was actually the strain that's used in the product, strain GHA, and not just some native strain. And in the process of doing this, we found only 9 non-target insects killed by Bottega. And these do include a few native plant hoppers like this one here in Canalinea conica, 3 of which killed 3 Flatyremus proxima killed by Bavaria bassiana. We found a couple Lepidopterans, 1 beetle, and 1 yellow jacket. But note that we never found any honeybees or bumblebees in these treated plots. And so at the same time this was going on at Norristown, we were also testing this Bottega product with some potted grapevines at the Penn State Berks campus. This work was done thanks to David Bittinger and some of his other workers at Penn State. And so this bar graph here, this shows you the total mortality 9 days after we sprayed. And we found that 2 rates of Bottega killed 99 to 100% of the adults directly treated. And then the residual treatment killed 57%. And this means that we sprayed the grapes and then we put the lanternflies on them 30 minutes later. So even just making contact with the treated surfaces does have some activity and kills about half of them. Okay, and now I'm going to switch more to some more biopesticide trials. These are done in our quarantine lab. The goals of these projects were to directly spray all the life stages with biopesticides and compare them to GHA, which was used at Norristown. And so this table, this summarizes these different products that we tested. So this includes Bioceres, Bottega again, Velifer, Naturalis, and PFR 97. And I wanted to note that these are all different strains of Bavaria bassiana. So there's GHA, but you can see these other products have different strains. And I've bolded some of these products So Bioceres, Bottega, Naturalis, PFR, these are all already registered to use outdoors on trees and grapes. Velifer, as of now, is only registered for indoor use, like in greenhouses. To do this, we need lots of lanternflies to spray. So we went down to the same field site every couple of weeks to collect them. We rear these on potted tree of heaven plants we grow in greenhouses, and then they're brought over to the quarantine lab, reared in cages like this. And for the bioassays, we then take these lanternflies, we put them into these cups, we spray them with the biopesticides with an airbrush, and then we transfer them to these potted tree of heaven plants. You can see I have these sort of smaller ones used for nymphs and larger plants that are used for adults. They have enough food. And then we check on these cages each day, look for these lanternflies. Some, like this in the photo, just die while they're still feeding, so they're sort of dangling from their mouth parts. And then cadavers are moved to these humid containers, which allows for fungi to grow out, and this verifies infections. So here's a bar graph. This is all of the data from the nymph bioassays. So first, second, third, and fourth instars. We have survival y-axis, days after spraying here. And what we found basically is that all of the Bavaria products work just as well as one another. This other product though, PFR97, not as good. It does kill them and it does actually grow out of them, so it's capable of infecting. But I never find this fungus out in the wild, so maybe it's just not as good as these Bavaria strains. And now we have the data for the adults in these bioassays. And again, Bavaria products working pretty well, killing like 80 to 100% of them after 14 days. But we found with the adults that actually this other product, Bioceres, this seemed to work best. This is a wettable powder formulation. Maybe it's just better at killing the adults, something about their physiology. Don't really know yet, but interesting that this product worked better than the other Bavaria products. And again, you can see purple line, this cordyceps product, cordyceps fungus in PFR, just not as good. And while doing this work, we noticed that some of these strains seem to produce more spores than others. Why is this important? Well, out in the field, especially later in the season, these adults start to aggregate and feed closer to one another. And we often see lanternflies feeding right next to these dead lanternflies with Bavaria bassiana. And so this opens up a window for spores to pass from one individual to another. And so we want to make the case that, well, maybe if you spray them with this biopesticide, you can get secondary infections. And so if it produces more spores, maybe it'll kill more lanternflies in the environment. So here, these are the canidia counts. These are the average number of spores per cadaver. And so we found that Bioceres does produce more of these spores on the nymphs compared to Bottega and Velifer. This Naturalis product just doesn't have as many spores on the cadavers. Then we have the data for the adult cadavers. Bioceres and Velifer now actually both producing a lot of spores on the adults. You can see, like in this photo, it's just covered compared to these photos here. So again, with the adults, Bioceres and Velifer did the best. Bottega Naturalis not as good for the adults. Okay, and then I'm going to talk about some experiments I did this summer in 2021. And so if you think back to the diversity data I talked about, describing all the different strains or genotypes, we saw that A, B, and L, these were the most common. And so we actually grew up these strains of fungi in the lab. I won't really go into how we did that. It was on barley flakes, and this was with the help of Stefan Juronsky. But basically, you just need to know that we produce the canidia or the infective spores. We are not using formulated products, so we're just testing the fungus itself. And we're comparing that to strain GHA. And this fungus was also grown up to produce canidia. So we weren't comparing this to Bottega. We're just testing the fungus alone. And so same methods as the other bioassays I talked about, spray them with the airbrush, put them on potted plants. So I'm just jumping right into the results for time's sake. But here's survival on the y-axis again. And so we find actually that strains A and L, very similar to the commercial strain GHA. This other strain we had, strain B, doesn't seem to work as well. We're not sure why, but even though it's common in the field, maybe it's just not as good at killing them, at least compared to these. But when I switch to the adult data, something a little different, kind of interesting, we actually see that strains A and L, they kill about 85 to 95% of the adults after 14 days, and they do kill them faster than the commercial strain GHA. This was only killing about, I think like 72% of the adults. And here it actually is similar to strain B now. So just jumping back, here's strain B on the nymphs. Now it's doing a little bit better against the adults. I should also say that we normally collect these strains in the field on adults. We only find infected nymphs kind of rarely in the field. They're also harder to find. The point here is that maybe these are strains that are better at killing adults than they are at killing nymphs. A lot of unanswered questions there, but interesting. And so I talked about spore production with the commercial products. We did something similar with the nymphs too. Here's a collage showing, this is what GHA looks like when it grows out of the nymphs. It grows out, but you can still kind of see the body parts, doesn't completely cover the bodies. You look at these wild strains in the field and they sort of blow up and they cover the entire body with Bavaria spores. And this is still ongoing. I still have more spores to count, but I did finish the third instar data. And here are, again, the conidia per cadaver. And we actually see that the better performing strains that killed the most lanternflies also produce the most conidia on the cadavers. So here's strain A, strain L with the most conidia, followed by strain B, and then the lowest produced by strain GHA. So pretty interesting, but I still have more counting to do. So to summarize the biocontrol studies, spotted lanternfly is still a very real economic threat, and it continues to spread. Poses very serious challenges to viticulture, ornamentals, and other industries. We have found now 4 different species of entomopathogenic fungi that infect them, and in some cases they can cause epizootics, especially with that back koa fungus. The diversity study showed that there are at least 20 genotypes of Bavaria bassiana infecting them in the field, and from those recent experiments, some show potential maybe to be registered in the future as some kind of biocontrol product, but we'd have to do some non-target tests to see how they affect other insects. And the currently available products like Bottega, Bioceres, Naturalis, they seem like they could be useful tools, but we'd have to do some more field studies to really see just how well they work in these outdoor settings. But I have some lingering questions from doing this work. I have received samples from people in Maryland, New Jersey, and West Virginia. So we know Bavaria bassiana is in these other states being invaded, but just this question of what other fungi will we find the more that it spreads out. Some modeling studies would be interesting to look at how you could predict epizootics based on population densities and rainfall in a given area. I have a lot of questions too about exploiting lanternfly behavior. They feed on different host plants throughout their life cycle. They have this sort of swarming and dispersal activity as adults, but maybe keying in on key time points in their life cycle to spray them. Like it seems like in September, October, they start to slow down after they've mated, and they sort of aggregate the bases of these trees. And so maybe treating them just before they lay eggs might be the best time to spray them, and also to hit most of the population at one time. But also I'm very interested in interactions between other insects in the environment, because we see so many of these yellow jackets in these areas. I made this goofy little Photoshop because I've actually seen Eurasian hornets, these larger hornets, pick up and kill yellow jackets to feeding over honeydew. So sometimes it seems like this honeydew production by the lanternflies is driving competition between other insects. But I'm also I'm interested in sort of transmission from one host to the other, because I see so many of these yellow jackets in the field, and we find them killed by Bavaria as well. And so it's this question of, are they passing it to the lanternflies, or are they getting it from the lanternflies? I don't really know. And this last fall season, I've been finding more of these yellow jackets killed by Bavaria, but still have to do the molecular work and confirm what's actually killing them. Okay, and I think I got about like 6 minutes, so I'm going to breeze through this last section. This is about a whole different study. No fungi here, but this was a very, very fun project, a lot of field work. This was carried out or led by Carrie Brown Lima. This is also with Angela Fuller, my boss, Ann Hayek, and some other people. We wanted to compare the efficacy of scent detection dogs to human searchers to see how well they could find lanternfly egg masses. And so we did this in 20 vineyards in Pennsylvania and New Jersey. So the colors there, those arrows, that just represents the different groups or clusters. They don't mean anything else, but they're sort of along different latitudes. So we have group 1 at the top, group 2 in the middle, group 3 mostly in New Jersey. We did this throughout January and May to do all this before the lanternfly eggs hatched. And so Carrie and Arden did the work of finding vineyards to participate. Arden did a lot of hard work to flag transects in and around these vineyards that we could search. We also searched the surrounding landscape. So this map shows you this one vineyard near Stroudsburg. So there's the 12 vineyard transects here, but we also searched along transects surrounding the vineyards to see where we'd find these eggs. And so, I mean, this was done for all of the sites with the forest and the vineyard itself. And, you know, you've probably seen photos of egg masses before, but just to show how well they hide, there's 3 in this photo. Boom, there they are on this cherry tree. They seem to like cherry trees. There's more than 50 on this photo on an oak tree, and boom, there they are, highlighted pink now. As a reminder, please don't ever move firewood, because you may not just have lanternfly eggs on them, you may also have like wood boring beetles on the inside. So never move firewood. And we do this with search teams. So like we have somebody that looks for the eggs, and we have somebody that records the data to speed up the process. Actually, in the wintertime when it snows, it becomes a little easier. Here's David on his back. You can call it the car mechanic crawl. And you can just glide along and find lanternfly eggs below these trunks while he searches. Sometimes when that snow melts, then you get these nice puddles to search in. So each day brought something new challenges every time we searched somewhere. Here's the scent detection dogs. There's Dia, there's Fagin, handled there by Joshua Beese. And so I just have some preliminary data. I don't really have time to go over all this, and we're still writing the paper. But this is being done by Ben Augustine and Angela Fuller. And so I think these dots are humans versus dogs. I'm not exactly sure, but these graphs on the left are showing sort of the detection capability. These solid lines are different humans. You can see human 3 here. I think this is David. So this showed that David, for example, was the best at finding egg masses in the vines. The dogs, for some reason, are not as good in the vineyards. We think maybe they're confused by scents of cadavers and other stimuli. But when you look at the forest down here, we see dogs are actually better at finding egg masses in these forested habitats compared to the humans. But just keep in mind this is preliminary. There's other data to show, but I just want to make the point here that, you know, maybe dogs would be better suited for the woods than for searching vines. And this is kind of my favorite slide to share. This is about what I call prime real estate, where to search on grapevines and other places. So there's the schematic showing typical vine structure. There's 6 egg masses in the picture. They're now highlighted yellow, and I'm sort of breaking down different areas to search. So one is to look at the undersides of cordons. You almost never see egg masses on the upper surface. They're almost always underneath. I think that the nymphs like to use gravity to their advantage. So when they hatch, they can just kind of use gravity to sort of fall out of the egg casing and crawl out. So there's some different pictures there, some easier to see than others. Tight spaces between trunks and supports. This is next. Some of these vines are sort of trained vertically with these support poles. And so lanternflies are seemingly good at like hide and seek. And so they will go right between these wedges and lay their egg masses here behind the vines. This image on the right, this was actually a wooden post in the vineyard, and it was cracked down the middle. And there were hundreds of egg masses on the inside of this post. So if there's a tight little space, lanternflies will crawl in there and lay their eggs. And when I showed this to the vineyard owner, I mean, he was shocked at just how many eggs were in there. And I think he destroyed or removed that pole and burnt it after seeing this. And then trunk bases, especially if there's like weeds or tall grass in your vineyard. So here's some pictures of that. You can see this egg mass here next to the dead adult. There's an egg mass there. This picture, the angle doesn't show it, but this trunk is sort of leaning towards the camera, and there's egg masses all up and down the base of this trunk. And then these support poles in the vineyards, like this site, they had a lot of tall grass next to their poles. And so lanternflies sort of use that to their advantage for cover, and they lay their eggs down there, down here. And then this was a site that had concrete posts, and these also were leaning at like a 30-degree angle. And so they seem to go for those leaning posts. Okay, I'm almost out of time, a few other slides, but just some patterns I saw. Check larger trees near the edges of vineyards, especially if they're like just uphill. This photo I showed you earlier, this is like a big oak tree, and it was just uphill of the vineyard at this site. And so this tree was just covered in just covered in egg masses. And this has kind of been already sort of verified by other researchers. This is Ashley and Heather Leach. They did a study looking at egg masses and also lanternfly populations. They basically found that most, like 44% of the egg masses, were found within 15 meters of the vineyard edge. So there's definitely some edge effects here. But you find fewer eggs sort of going into the center of the vineyard plots. Talked a lot about leaning structures. Well, if you have leaning trees around the property, look under there too. This was a vineyard, this cherry tree, it was maybe like 30 yards from the vines, just on the edge. This cherry tree is leaning over, and there's hundreds of egg masses under there. And also last, if there's any sort of stone or metal structures around the property, they seem to like laying their eggs on there too. So, look at cinder blocks, rusty metals, and so forth. In the process of doing this though, I've also seen a lot of mantis egg cases. I'm curious if they are sort of adapting to this new invasive and using it as food. You could sometimes even see lanternfly eggs right next to mantis euthecas. I don't know which one was laid first, but interesting that they're co-occurring here. And if you just search online for mantids, lanternflies, you see a lot of cool pictures like this. So again, curious in the future if we'll see these interactions become more common. But to summarize the vineyard studies so far, it seems like dogs are better in the forest or woodlots, but humans might be more effective in the vineyard transects. When the study is completed, when the paper comes out, this can help us to inform stakeholders on when and where to use detection dogs, but also some of these patterns of egg distribution can help growers to find and destroy these egg masses. Okay, so that's the end of my talk, acknowledging all the funding sources, all the people that helped, but I'm not going to name all one by one. I want to save some time for questions, and thanks for watching. I guess, Brian, if you're there, if you want to help me out with questions.
Wow, a lot for us to think about. I really appreciate all you shared with us. And yeah, did I catch there that you actually named that new entomopathic fungus there?
Yeah, so if you go to like Mycobank or something, you can see Clifton, Hayek, and Castrillo. There's Luella Castrillo down here. So the three of us are on the naming authority for that. So yeah, we named it, and we named it Ophiocordyceps delicatula because it shares the same species epithet as Lycorma delicatula. I didn't come up with any other cool nicknames, so we just went with that.
That sounds good. Yeah, so we do have some questions that are showing up in chat. And so you can take a look at those, or I'll read them out. One that came up early during your presentation was they're wondering how you determine whether a fungus killed the lanternfly versus a decomposing organism that might already be on a dead spotted lanternfly. And that was from Saul V.
Good question. Yeah, sometimes, you'll see a saprophytic fungus that looks white and fuzzy. I've had people send me samples thinking it was Bavaria. Really though, all you have to do is sort of surface sterilize the cadaver. So if you get a sample with what looks like fungus on it, you can clean it off with ethanol or some other chemicals, even like diluted bleach. And then you would plate that out in a container just to confirm that it's not a saprophyte. Otherwise, it's pretty apparent if you have what I call like a fresh cadaver or a lanternfly that has just died. We check on those frequently. So if we put it in a container, we can sort of see the fungus start to grow out between the leg joints, parts of the abdomen, between the parts of their cuticle. So we have to like see that the fungus is growing out of the body and not just growing on top of it.
Great. Another question is, have you tried these treatments on nymphs or just the adults, or maybe both?
Both. Yeah. So in those biopesticide trials, I did all life stages. Again, and in 2021, same thing. We tested third, fourth instars and adults. Yeah.
Great. And then Eric Smith asked, how often do the SLF oviposit, and does Bavaria bassiana affect the egg production ovipositioning before killing individuals?
Yeah, so we'd call those sublethal effects. Other pests do have sublethal effects from like Bavaria infection or other pathogens. So there's evidence that they'll reduce not just the number of eggs they lay, but the number of eggs that the female deposits. We haven't done that yet, looking at egg production or egg laying in lanternflies, but I imagine, if we see it slow them down before they die, I imagine it would have an effect. And before anybody else asks, like Stefan Jaronsky and a student, Jason Bielski at Virginia Tech, are looking at whether you can use Bavaria to spray egg masses, and then they're looking at what happens if those nymphs hatch out. Will they pick up the spores when they hatch and then die from those infections? But I think they're still finishing that study.
Cool, thanks. Amara Dunn asked, she knows that you shared the information in terms of insects being infected by Bavaria in your field trials. What else is known about the safety of Bottega or other biopesticides for bees or natural enemies? Right.
So before any of these products do get registered by the EPA, they do the safety tests on targets, especially honeybees. And actually, I've said his name, I'm gonna say it again, Stefan Jaronsky here, he's in this bottom left photo. Like, he actually had done some of these trials himself where they had to apply such a high dose of the Bavaria formulation that, this was a dose higher than you would normally spray, and they still had a hard time killing honeybees. And so it also depends on the pollinator you're talking about. Like honeybees, since they're a colony and they're eusocial, they will kick when they do get infected bees, they will kick them sort of out of the hive before they have a chance to sporulate and contaminate the rest of the beehive. So I would argue that they're pretty safe for those kinds of pollinators. And keep in mind that they groom each other. Like, people have tried killing ants, for example, with these products, and they have such a hard time because the ants are always grooming each other and protecting the colony. So I think the short answer is I think they're relatively safe compared to chemical insecticides.
Right. We're at that point where we still have a lot of people here, but I'm going to invite, have a couple questions that I think are probably best asked by the individuals who posted them, and it can be more of a conversation. So we're at that point in our seminar. So I'd invite HB, who posted a question about the fungus that you named, and they know a little bit about that and might like to share that. Feel free to unmute yourself, and we can have a discussion that way.
Hi, thank you. Can you hear me?
Yep.
Yes.
Hi, Hannah.
Okay, so it's Hannah Bradley. I was wondering about that new fungal species that you found. I agree, that's really awesome. You found a new species and named it.
Thanks.
I was just curious what you know about it from Asia. I assume, are you thinking it's not native, it's from Asia and came with spotted lanternfly? What do you know about the its effect on? Yeah. Well,
yeah, we don't we would argue that it's a native fungus because, I mean, in order for a fungus to move from lanternfly's native range, it would need to be a pathogen that was transported with the egg masses. So I think it's very unlikely that this is some fungus that came with the lanternflies initially. Ophiocordyceps, they do kill a lot of different insects. Well, not a lot, but they kill some other hemipterans. I would speculate that maybe it infected some other kind of plant hopper or tree hopper, like those flatoremids or something. That one was found in Lancaster, Pennsylvania, but I just never had the chance to go that site. I never had the chance to go to that site myself. That sample was sent from USDA APHIS workers. But yeah, sorry, I can't really say or know if it's native or not, but I would argue that it's a native fungus that's just jumped over to lanternflies.
Okay. Yeah, I was just curious because it's, newly detected, there's nothing known about it before. But I
Yeah.
guess there's not that much work on fungus of lanternflies, I
Yeah.
assume.
Yeah, I mean, our knowledge of entomopathogenic fungi is still pretty lacking. There's a lot of work to be done on just the taxonomy alone. In order to like know if it's really native, you'd have to go to like that site and collect as many insects as possible and detect that fungus in the population. So unless you're actually monitoring it directly, like I don't know how frequently, but often, like those fungi will sporulate in the wild and kill the insects, and then they will disappear and we'll never know that they were there. So
Mm-hmm.
my short answer is like we need to do more work to know for sure.
That totally makes sense. Thanks.
Yeah. Thanks, Anna.
All right, so I see Jay Watson and Greg Para have a couple questions. If you'd like to unmute, go ahead.
Hi, thanks, Eric. This is James Watson here.
Hey.
Just wondering, hypothetically, let's say we have a situation where we have large likelihood of naturally occurring Bavaria, let's say in the soils, and we get a situation where we have then large amounts of lanternfly that move into the area and their population builds. Let's say weather conditions are very conducive to that large population of lanternflies succumbing to Bavaria. I'm just wondering what the potential is then to see a spike in infections of native insects from the Bavaria as well, since some of these native insects like to hang out at these sites where there's lots of lanternflies.
Yeah. So if I understand your question, it's like, can we sort of predict Bavaria and other fungal infections, based on weather data, population data, but also like, what is there concern about native insects, right?
Right. In other words, is there a potential for having large amounts of spotted lanternfly that then become infected with Bavaria, leading to infections of native insects as well that hang around where the lanternfly are feeding and dying?
Yeah, that's kind of what I was getting at with the yellow jackets.
Right.
And so I think that's a term we'd call spillover, where a pathogen grows in abundance and so then it spills over and it infects other insects in the environment. A couple points there. One, about predicting it, we're still working on that. We call that epizootiology, or the study of epizootics. So we're still doing that. We did some data collection this year, sort of tracking to see when do we see more Bavaria or Bacoa, and why. It's sometimes we'll go and we'll sample like 50 adults from a site. We rear them in the lab and we just wait to see if they die. And sometimes we see like 20 to 30% that we sample in a given week are dying. And I believe that we kind of already know that, like when there's a rainfall event, we tend to see, more Bavaria infections. Two, I don't know so much about spillover, especially in areas that are invaded. If they're mostly tree of heaven, I don't see a lot of other native insects, feeding on tree of heaven in those areas. It's mostly just like a lot of little flies and yellow jackets, anything that goes to collect the honeydew. So if you're seeing an uptick in fungal infections, then definitely the other insects going to that area, like those yellow jackets, are probably at higher risk of getting infected. But also keep in mind that this is a new invasive insect. It's not already adapted to like the climate and the pathogens that are in Pennsylvania and these other states. So the native insects that have already been here have been exposed to native strains of Bavaria for a long time. And so they may already be more resilient to Bavaria than lanternflies are. Like they didn't sort of develop the immune system that the native insects have. So, you know, when I go to these sites, I mostly just find dead lanternflies, but to really thoroughly search all the non-targets being impacted, I mean, we'd have to scale up and do like pitfall traps, mist nets, all this other stuff. But currently I've just been like going and looking at, you know, ailanthus trees and collecting the non-targets that are near there. So it is somewhat limited by that. I don't know if I answered your question. There's a lot there, but
I think you answered it really well, and I appreciate the point about the possible resiliency to Bavaria infection of native insect populations.
yeah, and I like sort of trying to answer Hannah's previous question. Like, there's more work to be done. Like, I can only speculate, but I can't say for sure.
Right.
And Greg, Para, I don't know if you have anything else.
Yeah, hey Eric. Yeah, I was just curious because you talked about the secondary infection, which I've always been very curious about with especially with Bavaria bassiana, and because I don't think there was any let's say like the ground cloths or anything else set up further out or any monitoring from that treatment site at Norristown, if that's correct. So you say like you were someone at set distances beyond that to see if you were having effect beyond the treatment site, and then also like once you apply to an area, do you think or have any idea of if now there would be residual effect the next year, based on what you're talking about, looking in the leaf litter, like the following spring when you were first looking for it, you were finding infected individuals. So I'm just curious about like your thoughts on a carryover effect even for the next season, even if you didn't treat that site.
Yeah, interesting question. I think there might be, I can only speak for like Sinking Spring. There's a town where I normally go to collect lanternflies to supply my colony. And like the first year I started going there in 2019 or so, I would see some Bavaria, and then I think I saw more of it this year in 2021. I know it was a pretty wet year compared to 2019, but it seems like as long as there's a dense population in like Sinking Spring, it seems like more and more Bavaria infections are occurring at that site. And that could be sort of this residual buildup of inoculum in the environment or in the soil. So I think that's a possibility. There's also the question of, I know Penn State's been doing some more additional trials. I wasn't there to help, but they've sort of looked at how to expand their sampling. They've even like tried spraying adults in September, but then there's issues with adults moving out of the site. So then like, how do you really measure efficacy if you've sprayed them and then they go somewhere else? So that's a tough question. But with the residual buildup and inoculum, when it comes to GHA, like if you're spraying a commercial biopesticide, I mean, we already know that the canidia are only viable for so long in the environment. Most of the lanternflies that we killed at Norristown were killed by that strain. That's in the paper. I didn't have time in the talk. So we know it kills them as long as it's there. But in the process of doing all of the diversity studies, like I have never ever found GHA in a field site in a natural population. So that's probably a strength of the product, is maybe the product works well to kill the lanternflies, but it doesn't spread in the environment and sort of outcompete native fungi. That's sort of a positive for a biopesticide. And so, I don't see GHA outside of that Norristown project. So I don't think you'll see a lot of spillover with that particular strain, but I have not done the experiments with other strains like Bioceres. Like if I spray Bioceres, will I be able to find it the next year infecting lanternflies and other insects? don't know. Can't answer that, but I feel like we are starting to see some buildup of native Bavaria in some of these field sites. So like, if I were to go back to Sinking Spring in a few years, the question is, what's happened to the lanternfly population, and is Bavaria still killing them? I'll let you know when I go back next year, if I go
Great.
back. But
Great.
thanks
It'll
for
be good to hear an update. Shifting over, you showed some great images and showed what a challenge it can be to see the egg masses. So Dan Gilrain had a question, and Dan, I don't know if you want to unmute and ask that.
I just wondered if in your inspections in January versus May, was there a noticeable difference in the appearance of the egg masses or ability to detect them between the two times?
When I had more help, it got easier. The first few times we searched, we didn't have like data recorders, and so we might have. We also probably got more practice and got better at finding them. But no, when it comes to like visibility, like we've took notes on like if it was raining, if it was sunny versus foggy. I feel like it's not really an issue with the weather, but I will say, like, and it's just talking about the appearance, like the egg masses kind of look the same. They don't, some may lose some of the coating if it rains, but when you have direct bright sunlight, it might actually be more difficult because then you get more shadows. So like when you're taking photos, or like you're a professional photographer and you're doing wedding photos, right? Like they prefer cloudier days because then there's better light diffusion. So I find it easier to find egg masses when it's kind of cloudy outside. And so like, I'm not looking into sunlight. But that's really all I could say about like what's more or less difficult.
Thanks. That's great.
Yep.
Great. They are sneaky in where they hide their egg masses, it seems.
Yeah, tight spaces and leaning structures are definite musts.
All right. Well, thanks for everybody for staying on. If you have to go, that's understandable because we are at the end of our time. But if there's any last questions that anybody would, or comment, anyone would like to unmute, feel free to do that. If you're okay with that, Eric.
Yeah.
Can I ask a quick question, Eric?
Yep. I see it.
Hi, thank you for the nice presentation. One of the things I'm just curious is like, if you have to suggest to spray for the vineyard, what should be our best strategy? Should we target the eggs in the drought time, or should we do just a couple of sprays in the summer? And does that has anything to do with the weather? Because I saw in one of your spray in June, even the control one was going down after a week or so. So the human disturbance or anything really doing something like that?
Yeah, good question. That was about like the best time to spray Bavaria and against different life stages, and also about the control. So first off, in the control, Norristown taught us a lot of things. Some things didn't work or they failed. We learned that it's really hard to have the control and the treated plots right next to each other, because you spray nymphs, like a lanternfly nymph can crawl like 50 meters in a day or so. So we think that maybe there was interplot movement, which sort of nullifies the observed impact of the spray. We said that 48% were reduced based on visual counts, but it might have actually been higher than that if we could actually keep, the controls and the treated ones away from each other. But, we learned from that. As far as, and that was to answer why the control populations also went down. Regarding when to spray. So I have some reservations about spraying nymphs because they will be spread out on roses, bittersweet, tree of heaven, kind of all throughout the landscape. And so it becomes very difficult to hit or target every individual nymph in the environment. My opinion is that it might be better and easier to treat a population when they're at the adult stage, but I have to sort of think more about their behavior, because there's the early adults in like August. That's before they start to mate and fly more actively. Spraying them in early September might be troublesome because they might go on the move and then you don't know if it worked. But at some point after they've made it and they're sexually mature, that's the point at which they start to really go back to tree of heaven. They're almost not, you can rarely find them on other host trees in like late September and October. And egg laying occurs in kind of middle of October until the end of the season. So I'm of the opinion that it might be easier to spray the adults, let's say in like late September, early October, and try to kill the population before the time they start laying eggs. So that's just my opinion. You know, I would have to do more field studies to prove that theory or that hypothesis. But I think it might be better to do the adults than it is to do the nymphs. But good question.
Great. All right. Well, I think that about wraps it up. Getting a lot of thank yous there on the chat. So we really do appreciate you taking the time to share this really current work, and look forward to what's next.
Yep. Yeah. Thanks, Brian. Thanks everybody for jumping in.
Thanks.
All right. Take care.
Take care.
Bye.